Thank you so much. Also, thank you so much for the opportunity here, and I'll get started. So, as you heard, a PhD student in Lausanne, nearing the completion of my PhD. So, microbial communities are everywhere and associated with animals across the tree of life. A question that remains, though, is how have they evolved and how is their structure shaped? To ask this, we need to compare the gut microbiome of animals that are evolutionarily related across host species. And then we can ask what are the underlying processes that shape this diversity. A useful framework to look at this is phylosymbiosis, which describes a pattern where more closely related hosts have a more similar microbiome composition. This pattern has been observed in many animals, including mammals, rodents, and primates, and also in social bees. This describes a pattern at the level of the community, but these are driven by the individual members that make up the community. For example, there would be generalists that are found across different animals versus specialists that are only found in certain hosts. But the key question is, what are the factors, the microbial factors or host factors that really drive these patterns? An important process is co-diversification, which has gotten a lot of attention increasingly. And it's the case where the microbial phylogeny is congruent to that of the host, and more closely related hosts also have more closely related microbes. And this pattern has been reported in primates. For example, you see here the family of Bifidobacteriaceae, and they have host-specific lineages that are congruent to that of the host. And there was another study where they look at various different clades and found that 20% of these clades show signs of co-diversification. But what are the other processes driving the entire community? And how about other animals? So this is where I introduce you to the bees. They have a simple composition with around 20 bacterial species across 5 to 8 genera, compared to other animals, which have hundreds, if not thousands of species. They're very tractable to study. They have a consistent microbiome across worker bees, which is acquired when they emerge into adults and maintained by social transmission between individuals and from colony that leads to new ones. And these are distributed globally and have very well-known ecology. To set the context, social bees overall are found across 3 clades: the stingless bees, bumblebees, and honeybees. And from 16 studies so far, we know that there are several shared microbial members like Bifidobacterium and some lactobacillus species, Snoccosella and Gilliamella, which are found particularly in bees. And if you isolate lactobacillus, for example, from these different hosts, there are pretty deep diverging clades that are also host-specific, leading to this common idea that there was a common ancestor of bees that were associated with these microbes, and the microbes have then diversified within their host systems. However, our biggest challenge so far is that past studies have either been limited in depth because they relied on 16S amplicon sequencing, or in breadth because isolating genomes is laborious and it's difficult to cover the entire community this way. So in our study, we used shotgun metagenomics. I'm going to go over some interesting results, but you can check out the entire study in our preprint on bioRxiv. So we focused on honeybees, which are closely related to each other and hence comparable, and they share several behavior and ecological traits, but are also distinct in terms of their nesting behavior, colony size, bee size, and so on. We covered several colonies across India and Malaysia, thanks to our collaborators in Malaysia, who did a lot of fieldwork for sampling, and our collaborators in India, who hosted me in what was supposed to be a holiday, but turned into a very productive sampling trip. And I will go over the key results now. So we cover the 3 major clades of bees, including the cavity nesters, where we also find the western honeybee, Apis mellifera, which is very well studied, and the other clades, the giant honeybees represented by dorsata, and the dwarf bees represented by Florea and Andreniformis, which, being important pollinators in their habitat, have still been understudied. So we dissected the bee gut, so we didn't rely on fecal samples, but could look directly at the microbiome and used Illumina sequencing. After this, we assembled the reads into mags and annotated them at the species level using the GTDB database. So first up, we see that there are mainly just 3 genera that are really shared across the bees. So far, a core has been talked about in terms of the microbes that are found in the Western bee, but really there are several others that are found in the giant honeybee, for example, which is interesting and at huge prevalence, which might also be important in their function. Next, what is it that we see at the species level, really, now that we can ask that with metagenomics? We see that the composition overall is host-specific, with the dwarf bees really overlapping and actually clustering quite close to Apis mellifera. And also, in terms of the functional potential of the community, we see based on the abundance of KEGG ortholog profiles that they have also host-specific distribution. So next we asked if more similar hosts also have more similar microbiomes. And this would seem to be the case across social bees. But in fact, when we just look at the honeybees, this is not really the case anymore. And Apis mellifera and the dwarf bees, for example, which are the farthest from each other, share a lot of members. So there is really not this correlation between host similarity and microbiome similarity anymore. Yet we see host specificity, right? And what might be the processes underlying this? So if it were the case that there was co-diversification, we wanted to check this by looking at the phylogenies of mags from the prevalent genera. And what we found was that there was not any signal of co-diversification more than expected by random chance. And if you look closely at one of the phylogenies, for example, we see a lot of host-specific lineages, but this isn't really congruent to the host. And in fact, we also see several clades which have mixed like signs of host switches. How does the specificity of each of these species look then? So over here, you see each circle representing a different species grouped by the genus that they belong to, and the prevalence in each host shown by the pie. And immediately you see there are several specialists and several generalists. And what's interesting is that even within the same genus, where traits like affecting dispersal, like aerotolerance, sporulation, would be shared, there's a difference in the specificity of these species. And what might be important, so one pattern we noticed, which is cool, is among the different host species, we see that there are several specialists in Apis dorsata and mellifera as compared to the others, which mostly share their members with one of these others. And this could be due to various factors. For example, in terms of host ecology, dorsata is unique because it's highly migratory. Apis mellifera, of course, is always our special one. This is the Western bee, which is highly managed. It's been bred and it's been transported to different parts of the world over many years by us. Next, we asked, within each of these species, what might the strain level profile look like? So consider these generalists, for example. And this is an example of Bombilactobacillus mellifer, where we look at the overall SNP profile and we plot each sample based on an NMDS. And it turns out that this looks like a generalist at the strain level as well, which is what you would expect. But however, its neighbor, on the other hand, actually shows a host-specific strain level distribution. And this holds even across locations. And this indicates that there are species, for example, found in Apis mellifera, both in India and Malaysia, that share the same strain, but not with the other host species found in the same location. So these patterns that we've now seen set us up very nicely to ask the question of what are the important mechanisms and the processes for maintaining host specificity. Bees are a great model to ask this because, as you can see on the right, we can colonize Apis mellifera with specific microbes, and we have an ever-growing strain collection thanks to all of our collaborators. And another question is, what are the consequences of the differences in functional potential? For example, we see this species of Disconomonas, which is host-specific, mainly found in the giant honeybee. which harbors a huge repertoire of chymes in these polysaccharide utilization loci. And these chymes are important to break down complex carbohydrates, which are found in pollen, which is an important part of the honeybee diet. So finally, to leave you with the key takeaways, first is we see that the community is host-specific in terms of taxonomy and function. This is not just because of evolutionary reasons. We don't see phylosymbiosis or widespread co-diversification. Instead, we in fact see that the host specificity of closely related microbes can vary a lot. And also in different hosts, there are some which harbor a lot of specialists and others which harbor a lot of generalists. And this could have to do with the ecology of the host or other important factors. And all of this points to a story where there might have been a common ancestor who acquired the microbiota, but over time, there've been switches, losses, and gains that have resulted in the patterns that we see today, where there is host specificity, but it is not super consistent with the evolutionary history. Finally, I would like to thank all of our collaborators and all of these tools without which any of this analysis couldn't have happened. And thank you so much.